Okay, so a mutagen is something in the environment that can cause a change in a DNA sequence. Types of mutagens we've talked about are radiation, such as UV radiation from the sun, radiation from X-rays. There are chemical mutagens, such as pesticides, weed killers, chemicals in cigarettes, carcinogens that can cause cancer, that can cause mutations in cells that cause cancer, cosmetics, and other things like that. Those are all mutagens. They cause changes in the DNA structure of the cells. Another one was infectious agents. Infectious agents like viruses, bacteria, HPV, the human papillomavirus is one in particular that is able to go in and mutate the genes in a cell and cause them to become cancerous. H. pylori is a bacteria that can become mutagenic. That tends to be in stomach ulcers. 27 says carcinogens, which are cancer-causing agents. Carcinogens, processed foods, cleaning products are all examples of which type of mutagens. Those would all be chemicals, chemical mutagens. 28 said, what is the result of crossing over and when does it occur? Okay, remember we talked about crossing over back when we talked about meiosis? Crossing over is when your chromosomes are lined up in homologous pairs. Chromosome 1 from mom lined up with chromosome 1 from dad, chromosome 2 from mom with chromosome 2 from dad, and so on. The homologous chromosomes are paired up, and while they're paired up, they undergo what's called synapses, where they're able to cross over each other and exchange some genetic material. It's called crossing over. The crossing over allows them to exchange genetic material, so one of them is not totally a mom chromosome and the other one totally a dad chromosome anymore. They swap some of that genetic material, and it doesn't happen the same way twice. So this increases genetic diversity. The whole purpose of crossing over is to increase genetic diversity. Okay, so it increases genetic diversity. That's what keeps you from looking exactly like your sibling. Okay, crossing over, and then we also talked about independent assortment. The chromosomes don't sort the same way twice. So they don't always give all the mom chromosomes to one gamete and all the dad chromosomes to the other. They assort independent of one another. So with the independent assortment and the crossing over, there's a great deal of genetic diversity, which again is the main reason organisms do sexual reproduction. So when does it occur? Crossing over occurs during prophase 1 of meiosis. Prophase 1 of meiosis. Number 29 says, using the pedigree below, answer the following questions. We're going to identify the individual marked as X. Identify the individual marked with the X. Okay, but there's not one marked with an X, so we're just going to identify as many of them as we can here. This is an X-linked recessive trait. Remember, the circles are females, squares are males. If they're shaded in, that means they have the trait that the pedigree is looking at. So, if it is a recessive trait, we know that individual number 1 here, she would be little x, because she is showing the recessive trait. We know that this father here is going to have at least one big X because he is showing the dominant trait. And this again is like a test cross, so little x with big X question mark. So we have to look at the offspring to determine the other allele for him. He has this daughter here who does not have the trait, so she would have a big X, and then she would have had to have gotten a little X from mom because mom only had little X's to give. So we know that she would have to be big X, little X, and this daughter would also have to be big X, little X. These two sons are affected by the trait, so they would both have to be little x, little x. So Mom gave one of those little X's, and if they have two little X's, Dad had to give a little X as well. So now we know that Dad would be big X, little x. Okay? And then these would be little x, and little x, little x. And then all these, his wife and their children are all showing the trait, so they would all be little x as well. Okay. This daughter, we know, we just said that she has a big X because she's showing the dominant trait, but she also got a little X from Mom because Mom could only give a little X. Well, she married someone else who is showing only the dominant trait, so he is big X and a question mark. Okay, so they have children here. That all we know about these three children, the only thing we can determine is that they have a big letter X because they are showing the dominant trait. But then we had this son right here who's showing the recessive trait. So he would have to be little x, little x. That we said both of these parents are showing the dominant trait, so they both had a big X, but for him to be affected with the trait, he had to be little x, little x. So that means Dad also had a little x that he gave, and Mom also had a little x that she gave. So he would be little x, and they would be big X, little x. Both of them would be. And then it said, what is the genotype of individual 2, 3, so generation 2? The Roman numerals mean which generation. This would be generation 1, this would be generation 2, and this would be generation 3. So individual Roman numeral 2, 3 would be generation 2, person number 3 right there. And we just said that he would be big X, little x. They're using H's instead of X's, so he would be big H, little h. So big H, little h. He would be heterozygous, and we know that because he had to give that little h to this son right here that is affected by the trait. Number 30 says be able to read a karyotype, particularly know how to determine the sex of an individual. And remember, karyotypes are the pictures of the chromosomes. They're lined up by size from bigger to smaller. Chromosome number 1, there's a pair of those, one from Mom, one from Dad. So chromosome number 1 would be the largest. When we get down to the 22nd pair, those would be the smallest. The smaller they are, the fewer genes we would expect them to have. So we would expect chromosome number 1 to have the most genes and chromosome number 22 to have the fewest. And that 23rd pair, that is where you would determine the sex of the individual. That's the sex chromosomes. That's where you would find the X or the Y. You're going to need to be able to look at a karyotype and determine that individual's gender. If there is an XX, it would be a female. If it's an XY, it would be a male. Sometimes there may be a disorder where an individual ends up being just an X. That's still a female if there's just an X. Or if they're XXY, those would be a result of non-disjunction. And ended up with too few or too many chromosomes. But if there's an XXY, even though there's two X's, if there is a Y there, that individual would be a male. So anytime there's a Y chromosome, the individual would be a male. So make sure you're able to look at a karyotype, answer questions on that karyotype. Make sure that you can identify if there's a disorder. We would expect to see 2 chromosomes at each site, 2 chromosomes 1s, 2 chromosome 2s, and so on. And if there's more than 2 or less than 2, then there would be a disorder going on there, which leads us to question number 31, which says, which chromosome number does Down syndrome affect, and how so? Down syndrome is also known as trisomy 21. And trisomy 21s mean that there are 3 chromosomes at the 21st chromosome site. Okay, so that individual would have 3 chromosome number 21s. Instead of having the normal 46 chromosomes, they would have a total of 47 chromosomes. And that, remember, is a result of non-disjunction at some point. Anaphase 2 of either the formation of the egg or the sperm, there was non-disjunction occurred. You said that maybe the egg ended up with 2 chromosome 21s in it, and then when it was fertilized by that sperm that had its own chromosome 21, that resulted in there being 3 chromosome 21s. So Down syndrome would be trisomy 21. There are 3 chromosome number 21s, and it makes the total number of chromosomes be 47 instead of the normal 46. Number 32 says cancer is a product of a mutation that blank. So cancer is the product of a mutation that causes a cell to divide out of control. Remember, cancer is uncontrolled cell division or mitosis out of control. Number 33 says, why are mutations important in the evolution of species? Mutations are important because they add to that genetic diversity. If an organism has a mutation that makes them more likely to survive a change in the environment, then that allows the species to continue on when others may have been susceptible to that change, and it may have killed off some of those, but then the ones that had that mutation may have been more able to survive. So mutations are important to evolution of species because they increase the genetic diversity. Usually we think about mutation as being a bad thing, but it does increase genetic diversity, and sometimes there are cases where the mutation actually makes an organism more able to survive changes in the environment, and it becomes a good thing if it stays with that organism and is passed on to the next generation. So 34 says, what happens when a cell cannot repair the damage caused by a mutagen? Well, then that mutation will become a part of that individual cell's genome that will remain in that DNA structure of that cell. And that's the question part of the study guide. We also have Punnett square problems that go along with the study guide. We have to be able to work the different Punnett squares. So that will be the next part of our videos on the study guide.